Good day, everyone. Today we will be presenting our study entitled Investigating the Effects of Microwaved and Non-Microwave Distilled Water on the Hydroponic Cultivation of Mung Beans or Vigna radiata L. Conducted by Shania F. Alea, Chelo Nicole V. Esguerra, John Marie D.C. Hill, and Miga P. Solivar. To start, legumes play an important role in human nutrition because they are affordable sources of protein, vitamins, fiber, and complex carbohydrates. One example of this is the mung bean, which is widely consumed and valued due to its short growth cycle and drought tolerance. With the increasing demand for sustainable agriculture, hydroponic farming has become an alternative method of cultivation. Hydroponics is a soilless farming technique where plants are grown using water as the primary medium to deliver nutrients directly to the plant roots. In this study, we focused on understanding whether microwaving distilled water has an effect on the growth and survival of mung bean seedlings when grown hydroponically. This comparison is important because water quality plays a major role in plant development, especially in hydroponic systems where soil nutrients are absent.
The main objective of this research was to compare the effects of non-microwave distilled water and microwave distilled water on the germination, survival rate, and biomass production of mung bean seedlings grown in a simple hydroponic setup. Specifically, the study aimed to observe seedling growth over time, measure survival rates of seedlings, and determine which water treatment produced healthier and larger seedlings. For the methods, the experiment began with the selection of healthy mung bean seed. Any seed that were damaged or discolored were removed to ensure consistent results. The selected seeds were soaked in distilled water for 24 hours to soften the seed coat and promote germination. Once early germination was observed, the seedlings were transferred into a simple hydroponic setup using two plastic containers. Container A, which is the control group, contained 100 ml of room temperature distilled water. Container B, which is the experimental group, contained 100 ml of distilled water heated in a microwave at 800 watts for 1 minute, then cooled down to room temperature before use. Small holes were made on the container covers to allow the roots for the seedlings to grow downward and reach the water below. For our data collection and measurement, seedlings were monitored every 2 days and classified as either alive or dead. After 8 days to 10 days, the survival rate was calculated. Additionally, biomass analysis was conducted. Seedlings were carefully removed, rinsed and separated into shoots and roots, which were weighed using a digital scale. The total biomass was computed by adding the shoot and root weights.
Results tables and figures. During the early stages, both setups showed successful germination. In day 2, both setups showed root emergence. However, seedlings in container B exhibited purple cotyledons, which may indicate early stress. For day 4, leaf growth and root elongation were visible in both treatments. Day 6, container A showed longer stems and healthier seedlings. In container B, several seedlings turned dark green and one seedling had already died. For day 8, container A still had surviving seedlings despite some discoloration. Container B showed more severe deterioration with stems turning down. And finally, day 10, all seedlings in both containers had died, indicating that water alone was insufficient to support long-term growth in a hydroponic system without nutrients. For the biomass comparison, the biomass data further supported these observations. Container A, which contained the distilled water, has the shoot mass of 0.5 grams, root mass 0.1 grams with a total biomass of 0.006 grams. And container B with the microwave distilled water has shoot mass 0.3 grams, root mass 0.1 grams with a total biomass of 0.004 grams. This indicates that seedlings grown in non-microwave distilled water develop more biomass, particularly in the shoot system, compared to those grown in microwave water.
For the tables and figures, the first table was about the observations made during the germination and early growth of mung beans in two different setups, labeled Setup A and Setup B over a 10-day period. In Table 1, we track changes in cotyledon color, leaf growth, root growth, and visible tissues. On day 2, both setups showed early germination with leaf growth present. Setup A had green cotyledons, while Setup B showed purple cotyledons, indicating stress or pigment changes. Roots in both setups were still short at this stage. By day 4, leaf growth became clearly visible in both setups, and roots started elongating. Cotyledons in setup A remained green, while setup B became darker purple. On day 6, most cotyledons turned dark green and longer roots were observed, although some root caps fell off, showing possible handling or environmental stress. By day 8, root, root thickening was observed in both setups, but discoloration appeared. Some seeds in setup A turned brown or dark, while most stems in setup B darkened. Finally, on day 10, all plants in both setups were dead, with thin roots and beans stuck to the container. In Table 2, we summarize seed health. By day 10, both setup A and setup B recorded zero healthy plants, with all seeds classified as damaged or dead, confirming that neither condition supported long-term plant survival. The next table the next table focuses on the comparison of root and shoot development between setup A and setup B as shown in table 3. Setup A recorded a higher shoot mass of 0.5 grams, while Setup B had a lower shoot mass of, of 0.3 grams, indicating better initial shoot growth in Setup A. However, both setups showed the same root mass of 0.1 grams, suggesting similar root development. When total biomass was calculated, setup A had a slightly higher value of 0.006 grams compared to 0.004 grams in setup B. This indicates that although setup A initially supported better growth, neither setup provided conditions suitable for sustained development, which aligns the observation that all plants eventually died by day 10. Further discussion, the results show that while both water treatments supported initial germination, the seedlings grown in non-microwave distilled water performed better overall. Microwave water appeared to induce early stress as seen in cotyledon discoloration and reduced growth performance. However, the death of all seedlings in both setups highlights an important limitation that hydroponic systems require nutrients and sufficient oxygen, not just water, to sustain plant growth over time. Thus, water quality alone cannot ensure plant survival in hydroponics without proper nutrient supplementation and environmental control.
In conclusion, the study demonstrates that non-microwave distilled water is more effective than microwave distilled water in supporting the early growth and survival of mung bean seedlings. And for future studies, we recommend using nutrient-enriched hydroponic solutions, adjusting microwaving duration or temperatures, and improving the oxygen availability in the growing environment. But despite its limitations, this experiment highlights the potential of hydroponic farming as a sustainable and cost-efficient agricultural method when properly managed. So that concludes our findings in this experiment, and thank you for listening.